Alright, hello everyone. Again, sorry this is late, I had a bit of a busy day. So, today we're going to cover the topics for exam 2. Right, so, yeah, let's just jump right into it and get started. So, today, I am not going to be covering the practice exam, I'm just going to be covering certain topics that you will find in tomorrow's exam. Once again, sorry this is late, but in any case, I hope you all do well. So let's just jump right into it. So, starting with let's just start with water. So good old H2O. So we learned about water, right? And water is super duper cool. You want to know why? Because of these. And these, as you may know, are polar covalent bonds. So the cool thing about water just being made of entirely polar covalent bonds is that because it's so small and because it has these polar covalent bonds, it is able to have multiple different properties in which are very unique and very necessary for survival of life as we know it, right? So, also as you may know, oxygen has two lone pairs. So these lone pairs have these partial negative charges, and so we gotta have partial negative, so we have to have partial positive somewhere, right? And if you hear a cat in the background, yeah, that's just my little boy. He likes to bother me when I'm working, but you know, it's fine. So, anyway. Sorry, just one sec. Okay. So, looking at water here. Water has 4 places in which it can make hydrogen bonds, right? And that's what these partial charges are for. So once we have another water molecule, the oxygen of one is going to be attracted to the hydrogen of another, right? Because of those partial charges. So opposites attract, we know that. We learned that last unit, right? So because of this, water is liquid at room temperature. Despite being such a small molecule, it is liquid at room temperature. When it freezes, it's less dense, all sorts of things. And all of these unique properties lend themselves to the fact that water is entirely polar and has these partial charges on them, right? So there are 4 unique properties of water that we need to know, right? 1 is adhesion. 2 is cohesion. 3 is high specific heat. Jesus, I can't spell. And 4 is ice is less dense than liquid. So, these 4 attributes of water are incredibly important for survival of life as we know it, like I said. Like I also said, these things lend themselves to the fact that water has these unique properties of having 4 available spots for hydrogen bonding, yet only having 2 covalent bonds, right? So the cool thing about this is that we have all of these special properties, right? So adhesion is when water sticks to a different surface. Okay, let me look up a good example of this because all right. So when you're working with a graduated cylinder, if you ever get into a chemistry lab, if you ever work with one of these, as you may have seen already, is that this is a graduated cylinder, and here in the middle That is the water line. This giant portion here is known as the meniscus, right? So the cool thing about the meniscus is that this is a prime example of adhesion in water, right? So adhesion is when water sticks to surfaces that aren't water, right? So it acts like an adhesive essentially. So if you have a water bottle next to you, or if you're drinking a glass of water, you can go ahead and spill that against the wall, and you'll see that the water doesn't instantly fall to the ground or evaporate or whatever. It just kind of stays there for a little bit, right? Or if you're drinking a nice chilled glass of water, you might notice that some water droplets tend to stick to the sides. Well, that's adhesion. And in the case of the meniscus, the glass is holding on, the water molecules are holding on to the glass here at the side, so the most accurate measure, excuse me, sorry, the most accurate measure for the amount of liquid is here at the bottom, right? Just 1 second. So, so yeah, that's adhesion. When water sticks to a surface that's not water. So, when we talk about cohesion, cohesion is when water sticks to a surface that is water. And sorry, my cat is just so demanding of my attention right now. So let me look up another example. Okay, this is perfect. So this is an example of cohesion. So right here we have a bug that is skating across the water, which shouldn't be possible, right? The only time we hear about people walking on water is, in the Bible. So, when we have this bug here, right, you see this bug is also walking on water. So what's going on here, right? This all lends itself to the cohesive properties of water. So if you've seen my Greek and Latin suffix um, handout, kind of resource rather, we know that co means with, right? So cohesion with water, cohesive to itself, right? So water has a special property. So let's say this is like a wave or something. So water has like a special property in which when something heavy lands on, or not even something heavy, when something lands on it, right? Let's say we have like a person. When something lands in the water, all the hydrogen bonds that are constantly being formed and broken and formed and broken, that still exists on the surface, right? So at certain heights, all of the surface tension that water creates with itself, eventually, if we have a person landing from high enough, they're going to, this water is essentially going to feel like a pile of concrete, right? Not good. However, stuff like this can happen because of the cohesive properties of water, right? Water just being able to stick to itself. It's being able to stick closely to itself, which is surface tension, right? So another really important thing about water is the high specific heat. So specific heat, it basically is a measure of how quickly something heats up, right? How quickly you could raise the temperature of something. Um, more specifically, more specifically, specific heat is the measure of how much energy it takes for something to heat up, right? So water has a specific heat of around 4.8, and I forget the units. It's like joules per gram cubed. I don't even know. But water has a very high specific heat, very very high. Something like metals, metals always have like maybe like below 1, right? So metals heat up really quickly, as you may know. So metals are super good for like conducting heat, conducting electricity. Water is very bad for both of those things, right? So if you've ever tried to boil a pot of water, you may notice that it takes freaking forever. It takes so long, and especially if you get into a chemistry lab, boiling water is going to be one of the most tedious activities you will come to find out. So, yeah, boiling water, not fun. However, high specific heat is very, 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 very important for the survival of life, right? Why is it important that we have high specific heat? Well, if I was made of alcohol, so alcohol is or drinking alcohol is C2H5OH, right? So it's a molecule that's a little bit bigger than water is, and it's far less polar with just the one OH group at the end, right? So if I was made of drinking alcohol and I went outside, I would not last very long at all, right? Eventually, I would just evaporate. I would literally evaporate because of how much quicker the heat is transferred in ethanol, drinking alcohol, than it is in water, right? Water has this incredibly high specific heat that allows it to resist temperature change, right? So that's incredibly important for life, right? That's very important for life. So yeah, that's just something that's very important to emphasize. I mean, like look at temperatures in New Mexico. I mean, like there's like a 20 to 30 degree difference between the highs and the lows, right? Today was like 80 and tonight's going to be like 50. You know, compare that to California and it's like a 10-degree difference because there's a lot more moisture in the air. It takes a lot more heat. It takes a lot more for the heat to dissipate and to be absorbed, right? Because there's more water in the air. There's more moisture because it's on the coast, right? So that's high specific heat. Just how much energy it takes for something to heat up. And water has a very, very high specific heat. So as for ice being less dense than liquid water, right? That's important because if you look at the Arctic, right? Stuff can live underneath ice, right? There's just like a giant sheet of ice, but there's stuff living under it as well. And that's because ice is less dense than liquid water, right? So if you ever have like a drink, right, and ice is floating in your water or in whatever, ice is floating, this is why, right? Let's discuss. So water normally has like this 107-degree angle. For the sake of simplicity, I'm going to draw with a 90-degree angle. So water, when liquid, just constantly breaks and reforms hydrogen bonds. But water, when solid, will keep these hydrogen bonds suspended. And it will create a lattice. It will create a lattice structure, right? And that is something that's that is something that is incredibly unique about water, right? That when frozen, these hydrogen bonds will be suspended, it will create a lattice, and it'll have more space. It'll have more space between the molecules than it does normally, right? And that's why ice is less dense than liquid water. So, let's review, right? So, coastal climate, like in California, because of the high specific heat, because of the high specific heat, coastal climate like California is moderated by that property of water, right? So it's incredibly important for that, right? And as for cohesion versus adhesion, cohesion is when the attractiveness of water molecules applies to itself, right? So water molecules are attracted to each other, right? They want to stick together because of those H bonds. Versus adhesion is when water wants to stick with onto other surfaces, right? So and we just talked about ice, right? So the water molecules spread further apart in ice, right? Which makes sense. That makes sense, right? Because we just talked about that. Is that clear? I can't hear you through the screen, but I hope that is clear. So these are the 4 properties of water that you need to know. So now that we're done talking about water, let's go ahead and talk about. Sorry, my cat has trapped my hand. So if you'll excuse me one moment. So now we talked about water. Now let's talk about pH, right? So the cool thing about pH, right, is that when it was first coined, when this phrase pH was first coined, this P didn't actually mean anything. I think it's hilarious. This P didn't mean anything. So that's a fun fact. You don't have to know that though. So pH is a logarithmic measure it's a negative log of the concentration of protons in solution. So that's more of a chemistry focused question. That's just what pH is measuring. So ultimately, if you see pH, we are measuring protons, right? And before we continue, I think it's important to note why they're called protons. So, we have hydrogen. Hydrogen has an atomic number of 1, is the first element, has an atomic weight of around 1.008. Great, that's cool. Hydrogen does not have any neutrons. Just by itself, if we worry about isotopes, then it does. Don't worry about that. We're not worrying about isotopes. Hydrogen by itself just has 1 proton and 1 orbiting electron, right? So if hydrogen has an electron added to it, which can happen, then this is called a hydronium, right? So that's H minus. Not quite as common as when hydrogen loses an electron. So when hydrogen loses an electron, it's H plus, which makes sense. We know that, right? H plus. But look at what's left behind when we lose that electron. When we lose that electron, look at what's left behind. Nothing. It's just a proton, right? So when hydrogen is ionized, when it loses an electron, what's left is a proton, right? So that's why we call them protons. That's why we call H plus a proton. So when we're measuring the acidity of something, when we're measuring the pH, we are measuring how many of these are in a solution, right? So, the pH scale, and it can go beyond 1 to 14. We just use it for simplicity's sake, and for not making us go crazy, because pH is a measure of the concentration of one solution, but not necessarily the concentration of another one. That doesn't make any sense. If you want to measure if you really want to measure the acidity of something, of a substance intrinsic acidic properties, you measure its pKa. But we're not worried about pK because that's chemistry related. We're worrying about pH, right? And pH talks about the acidity of a solution, right? So when we're talking about the acidity of a solution, we talk about a scale. A scale from 1 to 14, with 7 in the middle. So 7 is like biological pH. That's like the pH of deionized water. 7 is like the middle point. It is neither acidic nor basic, right? So 7 is like this kind of Goldilocks zone for living things, right? We definitely don't want to get too acidic, and we definitely don't want to get too basic, right? That's for living things anyway. And once we go down, right, 6, 7, 8. Sorry, what am I talking about? 7, 6, 5, 4, 3, 2, and 1. We're going down on a scale of exponential, right? So 6 is 10 times more acidic than 7, and 5 is 100 times more acidic than 7, and 4 is 10 times more acidic than 5, etc. Right? So you don't really need to know that. I just had some questions regarding that. It's an exponential scale, right? So as we go more this way, as we are gaining more protons, we are getting more acidic. As we gain protons, we get more acidic. Sorry, no, I wish I could have done that better. Okay. More acidic. There we go. So as we're going this way, we're getting more acidic. And as we're going this way, we are losing concentration of protons. So we are getting more basic. More basic, right? So like I said, 7 is like a sort of nice in between where we're not acidic and we're not basic. We have a nice balance of both, right? So that's a pH scale. So over here on 1, like 2 is like lemon juice, so that's like citric acid, and then 1 is like battery acid, which is like sulfuric acid, and which, like I said, pH can change depending on the concentration of something. So lemon juice can actually be a scale of 1, just depending on how concentrated the lemon juice is. Whatever, right? That's just like an example. And as we go more this way, as we get more alkaline, more basic, that's just another word for basic, alkaline is, we have like bleach and like detergents and whatever, right? So that's more basic. Right, so like I said, we want this nice sweet spot in our bodies. Our physiological pH is like 7.4. And that's really important for amino acids, for nucleic acids, for all sorts of things in our body. Just maintaining that pH is incredibly important. Right? So yeah. This scale, and write this down, this scale is a measure of the concentration of protons. pH scale is a measure of the concentration of protons in solution. Write that down, please. If you're going to remember anything about pH, write that one down specifically. Right? So as we go left of 7, we're getting more acidic. Right of 7, we're getting more basic. We're changing the concentration of protons in solution. Right? So if I say I have a bottle of water with a pH of 7, and then I have a bottle of water with a little bit of lemon juice in it with a pH of 5, right? Which one is going to be more acidic? The one with 5, right? Because it's going to be to the left of 7. How about if I have a bottle of water with a pH of 9 and then a bottle of water with a pH of 8? Which one is going to be more acidic? The one with 8, right? Because despite both of them being basic, one is less basic than the other, right? So that's just yeah. So that's just the basics of the pH scale, which a lot of people, and myself included, when I first understood pH, is quite confusing. So please, what I just discussed, please just worry yourself about that. So let's move on. So we talked about water, we talked about pH. So now we get into biological molecules, right? And biological molecules are super cool. We're going to talk about macromolecules. It's going to be great. You're going to love it. I promise. So let's do it. So before we talk about biological molecules, we have to talk about the functional groups, right? So the functional groups are super duper great Most of them are, anyway. Some of them are really stupid and dumb, and you shouldn't know them. Let's talk about the ones that are really stupid and dumb. This is a sulfhydryl group. If you've attended one of my sessions, or if you've heard me talk about sulfhydryl, you will know that I have a lot of beef with sulfhydryl. I have a lot of beef with sulfhydryl. I think sulfhydryl is stupid and dumb and not something we need to know. And here's a secret between you and I. Sulfhydryl is not something you need to know for the exam. You heard it here first, folks. You do not need to know sulfhydryl groups for your exam. So please, whatever you do, if you get anything from this video, do not study sulfhydryl. It's pointless and stupid and dumb. Do not pay attention to this. Do not study sulfhydryl. That is the worst functional group out of all of them. They're stupid and dumb. So, let's get into the real functional groups that you do need to know. Right? Starting with arguably the most important one. So when I write R, that's just referring to anything that's not hydrogen. Right? So R could be a carbon, R could be another oxygen, R can be a nitrogen, R could be anything. Right? It's all in your imagination. R could be whatever you want it to be. What we actually care about is on this side. OH. So what is this? This is a hydroxyl group. Otherwise known as an alcohol group. You could hear it both ways. On the exam, you're definitely going to see hydroxyl, but in the future, you will see it either hydroxyl or alcohol. Especially in organic chemistry, you will see it as alcohol, but we're not worried about alcohol yet. Right? We're worried about hydroxyl. So, hydrogen, oxyl, oxygen, OH. Simple. Easy to remember. Hydroxyl, OH. And now what's special about this? It's polar. That's pretty much it. It's polar. This group allows something to be polar, right? So take the example of this is ethane. So my name is Ethan. Now this is ethane. So the thing about ethane is it's a gas at room temperature. Right? It just evaporates. You could burn it, whatever. Ethane only has 2 carbons. But the thing about ethane, right, is that when we remove this H and put an OH, now this is ethanol. So because we added this hydroxyl group, this all of a sudden is polar because ethane is nonpolar because it's a hydrocarbon, right? Now, when we add the hydroxyl group, it becomes an alcohol. It's an ethanol. Now it's liquid at room temperature, and we could drink it and have good times and parties and whatever you do with alcohol, right? Or use as a solvent if you work in a lab. Yeah, all because of this OH group. It gains all sorts of different properties when you add this one group that's polar. And that's the cool thing about hydroxyl groups, right? It just adds a polar group to a molecule. Hydroxyl. So now, let's get into one that's not nearly as important, right? And that's the carbonyl. So, I'm going to draw this kind of carefully. Notice how I didn't put R groups here, because we're going to discuss that in a second. So this is a carbonyl, or carbonyl. Carbonyl is more correct if you're really pedantic like I am. So carbonyl can be structured into 2 subcategories, and that is a ketone or an aldehyde. So notice how I didn't draw this C here. It's just this is just the shorthand called the bond line structure, right? So when you see an apex, when you see a corner or an apex, that's just an indication there's a carbon there. So this is a ketone, and this is an aldehyde. So what's the difference between these two? Well, the similarity between them proceeds not to answer the question. The similarity between them is that we have this carbon double bonded to an oxygen, right? So the difference between ketone and aldehyde is that a ketone is in the middle of a molecule. Notice how it has two R groups, one R, one R prime, right? And then an aldehyde is at the end of a molecule. Notice how it has an H. So that's really the only difference. What is supposed to be emphasized, however, is that there's this double bonded carbon to an oxygen. So you really only have to know what a carbonyl is and recognize that both of these are carbonyls. But a ketone is not an aldehyde. And not all carbonyls are aldehydes, but all aldehydes are carbonyls. And you can say that about ketones. Not all ketones are carbonyls. Not all carbonyls are ketones. Just remember this double bonded C to an O, and you're golden, right? So the thing about a carbonyl is that once you add an OH, this becomes a carboxyl. So I don't really like the name carboxyl. It's not really specific as to what it can do. While technically correct, carboxyl is correct. These are more commonly known as carboxylic acids. And that's for the future, right? That's for the future. But it helps to know that these are in fact acids, because these are going to come in handy when we talk about amino acids, right? This is the acid portion of amino acid, right? We can say carboxyl, but I always refer to these as carboxylic acids. So the thing about this is that this is in fact an acid, right? So an acid is simply defined as something that can lose a proton in solution, right? So this proton is just begging to leave, right? So because of that, we can say that this carboxylic acid can now be deprotonated, which is the name for something when it loses a proton, and become negatively charged. Like I said, super important for amino acids, right? So now this is called something different. This is called a carboxylate. You don't need to know that. It's just for the curious among you. Just know that a carboxyl group can be, and this goes for nearly every single carboxyl group, is that because it has this OH at the end, it could lose that H and become deprotonated and become negatively charged. So this is a carboxyl group. Carbon, oxygen, carboxyl. Or more specifically, carbon and two oxygens, carboxyl. Right? That one's super duper important. So while we're on the topic of amino acids, let's talk about the amino group. So the amino group looks something like this. Right? So amino groups are NH2. So amino. And amino is only something that you see in biology. Everywhere else, this is called an amine, with an E at the end instead of an O. Everywhere else, you will see amine. But here in biology, they're called aminos. So the cool thing about aminos is that, similar to carboxylic acid, these are present in, well, amino acids. But the thing about this is that it could also become charged in solution, right? But the unique thing about amino groups, okay, maybe not unique, but the thing about amino groups is that it could actually be protonated. It can gain a positive charge in solution. So, what does that mean? It means that it is a base. Amino groups act as bases. They act as proton acceptors. So when something accepts a proton in solution, it is a base. When something loses a proton in solution, it is an acid. So the aftermath of a base is that it becomes positive, and the aftermath of an acid is that it becomes negative. So, I hope that was clear. In any case, this is what's happening with an amino group, which once again we're going to look more of this in later when we talk about amino acids. Just what's happening here with the protonation and the fact that it's becoming positive. Right? So, yeah, those are your functional groups. And like I said about the carbonyl group, you really only have to recognize the two carbonyls. You don't have to know the difference between an aldehyde and a ketone. You just have to know that they're both carbonyls. I hope you understand. Because once again, I can't see you through the screen. I wish I could, but anyway. Functional groups are not too emphasized on the exam, right? There's just something that you're going to need to know for later, right? And arguably, along with OH, arguably the most important functional group that you're going to see in this class is the phosphate group. Now, I love the phosphate group. The phosphate group is like the anti-sulfhydryl, right? It is incredibly useful. You see it all the time, and it is very, very important for cell signaling, for cell function, for energy usage. This is the phosphate group. The phosphate group is so important because you see the phosphate everywhere. You see it in DNA, you see it in ATP, you see it in phospholipids, you see it everywhere because phosphate is so important. So, let's discuss. A phosphate group is negatively charged, right? As you can see here on this O and here on this O. And I hear you might be screaming through the screen, Ethan, what the hell are you doing? That atom has 5 bonds on it. Well, I have this for you. Phosphorus can actually make 5 bonds, despite only having 5 valence electrons. It can make 5 bonds. When normally, something like nitrogen, just one element up, can make only 3. Right? So what the hell is going on? Well, that's a more chemistry-related question. Just rest in the fact that this phosphorus can indeed make 5 bonds. It is very unique in that sense. So phosphate, you just have to know that phosphate groups change molecule conformation. Okay, this is going to be for later. You don't have to know this now. Just know that phosphate groups are very important because you're going to see them in phospholipids. I just love phosphate groups though. It's just that they could change the function of a certain enzyme. They're carried out in certain in ATP and energy molecules. It's just phosphate is incredible. So the thing about phosphate as well is that sometimes we don't want to draw all of this nonsense, right? As much as I love phosphate, it's a pain in the ass to draw. Let's be real. So the thing about that is that we could say whatever it's attached to, and then shorthand it to a P with a circle around it. And for whatever reason, phosphate groups are almost always depicted as being yellow. Don't ask me why, that's just how it is, right? They're almost always yellow. So you can always shorthand all of this, into just this. So, yeah, that's the phosphate group. So, those are all of our functional groups. I hope you're all feeling well and dandy and fine with all of that. Right? Because functional groups is something that a lot of students tend to struggle with, and I'm sorry to hear that, but it just is the case, unfortunately. So, with all that being said, let's move on to the macromolecules. Right? So, with the macromolecules, just give me 1 second. Yep, with the macromolecules, we have 4 that we care about. We have carbohydrates. We have nucleic acids. We have proteins. And we have lipids. So these are our 4 macromolecules, right? All of these perform different functions in our bodies with different effects, right? And we sort of divvy them up like this because lipids are kind of in their own class of weird, right? Carbohydrates, nucleic acids, and proteins all have the property such that they are essentially beads on a chain, right? So these all have monomers. All of these have monomers. Essentially meaning that we have one singular item, and when we put a bunch of those items together, they create a polymer, and a polymer performs a different function than a monomer, right? That lipids do not have monomers. They kind of just exist on their own, and they do their own thing. They're in some sense their own monomer. So, but we will discuss lipids a little bit later. Don't you worry. So, let's talk about sugars. So sugars always have the basic structure of C H2O. And we can put this around parentheses with an N. So when this N is equal to 2, for example, we just multiply everything in the parentheses by 2. Right? So that would be C2H4O2. Right? Or in the case of glucose, we could say N equals 6. We have C6H12O6. That's glucose. But of course, there are other isomers. Yeah, there are other isomers of C6H12O6. That could be something else. Don't worry about it. Just know that this is the basic chemical structure for a sugar. So what does glucose look like? Glucose can look like 2 different things. It can kind of look like a straight line. We don't ever worry ourselves with a straight line conformation. So you will most often see a sugar kind of in this, what's known as a Hawthorne projection. Right? Where we have an O here in the corner, and then we have a branch that leads to an OH, and then we have a bunch of OHs surrounding it. This is glucose. So we have 1, 2, 3, 4, 5, and then if we go right one more, that's an O. We go up 6 carbons, that's a sugar molecule. Great. What do we do with that? So, I'm going to say this with caution, right? Because you don't need to. Here's another secret between you and me. We don't really need to know a whole lot about sugars. In previous years, you had to know a whole hell of a lot more, but now you don't really need to know a lot, right? Just know that this is one monomer of sugar. This is one glucose molecule. Glucose, right? So you will see this, you will at the very least see this on your exam. One glucose molecule, right? Just have to know the general structure of what glucose or what a sugar may look like. Just has a bunch of OHs surrounding it. There's probably a ring. There's almost always going to be a ring, because if there's not, then anyway. So yeah, that's glucose. So if you put glucose in a string, you put a bunch of them in a string, you can get a couple of different things, right? So like I said, I'm going to say this with caution, but you don't really need to know alpha versus beta glycosidic linkages. You don't need to know 1,6 glycosidic linkages. It's not super necessary. Once again, that's a secret between you and me. At some point, you just gotta look at the content you've been given, and you just have to say, wow, that is a lot, right? And sugars are not really something that you should stress a whole lot about. Sugars are super important, sure, but they could be a lot, right? Especially with beta 1,4 or alpha 1,4 or alpha 1,6, you don't need to know any of that, right? So don't even stress. Don't even stress. But there's something I do want to emphasize about carbohydrates, right? And that is the type of that is the type of polymers that they can make. So when we get a bunch of glucose in a straight line, a couple things can happen, right? We can get three types. Sorry, 1 second. We can get three types of molecules, right? Three types of sugars, and that is starch, glycogen, and cellulose. And there's a couple of, I think I spelled that wrong. Cellulose. There we go. And there's a couple other ones that you don't need to know, like peptidoglycan. That's a sugar. What is it? Hell if I know. You don't need to know it. And then there's also chitin, I think. Yeah. These ones, you don't gotta know them. But the big three, starch, glycogen, and cellulose, which I'm gonna cross these out because you don't need to know them. Starch, glycogen, and cellulose, those are the big three. You do have to know these, right? Because they're super duper important, right? You eat a starchy meal, you get a lot of energy, right? You eat a whole big old pile of spaghetti, that's very starchy, right? If you're carb loading the day before a big run or the day before a big event, that's starch. That's quick, accessible energy that your body can use. It could just break off single molecules of glucose. That's starch, baby. And the same thing with glycogen. Glycogen is just like starch. They're like damn near the exact same thing, right? So let me show you a picture of starch and glycogen. Like I said, they're like almost, starch and glycogen are like almost the same. Like they might as well be the same molecule. Let's be real. Let's not kid ourselves here. They're so incredibly similar. I'm not sure why they're different or what makes them different. I think one has a little bit more branching out than the other, but really it's not something that you need to worry yourself about. Really, I'm trying to look out for you guys here. This is a starch molecule, right? And now that I'm looking at it, I drew the glucose wrong. Whatever, that's fine. So the thing about starch, so this is the thing about 1,4 glycosidic linkage, right? Versus and 1,6. So like I said, we have this glucose molecule over here. We count 1, 2, 3, 4, 5, 6, right? So we go clockwise. So 1, so we have So, for example, let's take this glucose. We have one on this sugar, and then the other sugar we have on the 4 carbon. So when we say 1,4 glycosidic linkage, glycosidic referring to sugar linkage, right? Glyco meaning sugar, meaning glucose, right? 1,4 at the 1,4 carbon position and the alpha position. So this is a alpha 1,4 glycosidic linkage. But here's the secret, guys. You don't need to know the reasoning behind that. You don't need to know the reasoning behind that. You just have to know that in the case of starch and in the case of glycogen, what holds those together are alpha 1,4. Or just more simply, just alpha. Right? These, both starch and glycogen, are alpha. Cellulose is beta. Right? So as for posterity's sake, let's take a look at glycogen as well. Right? So this is a MCAT study guide, whatever. Yeah, same thing here. Glycogen is super duper branched out. And because it's not as organized as cellulose, it doesn't pack nearly as well, meaning that we can actually digest glycogen and use it for energy storage, right? So guys, listen to me here. Alpha 1,4, alpha 1,6. That's the thing about glycogen. It has alpha linkages, alpha glycosidic linkages. Listen to me. Alpha glycosidic linkages. That's all you gotta know about sugar. That's all you gotta know about it, right? Starch and glycogen have alpha, and then cellulose has beta. You don't need to know the orientation difference. You don't need to know any of that BS. You don't have to know any of that, right? All you have to do is bring your butt to the exam and be able to say that starch and glycogen have alpha glycosidic linkages and cellulose has beta. So, like I said, for the sake of posterity, I'm going to show you cellulose as well. This is cellulose. So notice, right, how we go up, and then we go down, and then we go up, and then we go down, and then we go down, and then we go up, and then we go down, and then we go up. That's because it has beta linkages. And because it goes down and up and down and up and down and up, just like this on a smaller scale, if we take another cellulose molecule, and another one, and another one, and we take a bunch of those because this is like just a few angstroms in width, we have a bunch of hydrogen bonds. And this is why we can't eat a tree or eat grass or do anything with cellulose, because it just goes right through us. We can't digest any of it. We can't utilize it for any actual energy. So that's the thing about sugar, right? Sugar has three primary functions. The third one we don't really care about quite yet, right? But sugar can either be for energy storage or just for energy in general, right? For quick use energy for the case of monosaccharides, or it could be used for energy storage in the case of starch and glycogen. Or, number two, it could be used for structure in the case of cellulose, and as I've crossed out here, it could also be used for peptidoglycan and chitin. Chitin is found in the exoskeletons of certain bugs and whatnot, right? Yeah. And then the third one is that sugars can be used for cell signaling. But we're not there yet, so don't worry about that last one. So, what did we learn about sugars, right? Sugars are super important. Yeah. You have monosaccharides, you have disaccharides. Di means 2, mono means 1. So if you see a monosaccharide, you should see 1 sugar. If you see a disaccharide, you should see 2. And if you see an oligo, that means you should see few. If you see polysaccharide, you should see many, right? So guys, listen to me. As long as you know that glycogen and starch have this alpha glycosidic linkage, you're good. That's all you gotta know. That's all you gotta know. The difference between glycogen, cellulose, or starch and cellulose. All you gotta know is that one is alpha, the other one is beta, right? And that does it for sugars, because sugars can get quite complicated. Sugar chemistry is a field that I would not touch with a 10-foot pole, because I don't know, it just doesn't interest me quite as much as other things do. But I mean, if you really want to get into sugar chemistry, you can do that. Not my cup of tea. You gotta learn a bunch of different conformations and a bunch of different ways to look at molecules, but it is an option. I mean, we do have all these artificial sweeteners. Maybe you could develop something. Maybe this video is directed towards someone who will develop something that doesn't cause cancer, like aspartame. Aspartame causes cancer. Don't make the next aspartame. Make the next something cool. Make the next, I don't know. Artificial sweeteners are a whole different bag. So, let's talk about nucleic acids. Nucleic acids have three. They have three. Sorry, I don't know why I totally lost my train of thought. They have three compartments, three sections, three portions, three parts, whatever you want to say. It has a sugar, a 5-carbon sugar. It has a nitrogenous base. So I'm just going to draw something basic here, just like, I don't know, something nitrogen containing. Let's say this has like a double bond here, and like there's like a I don't know. This is a nitrogenous base. Don't worry about what I just drew. And then we have the phosphate. This is your basic nucleotide. Nucleotides make up DNA, and they make up RNA. Now, I hear you might be asking, what the hell is the difference between DNA and RNA? Well, allow me to tell you. RNA stands for ribonucleic acid. Okay, simple enough. DNA stands for deoxyribonucleic acid. See the difference? Ribonucleic acid, deoxyribonucleic acid. So I'm going to go ahead and copy this, move it over here, and I'm going to show you why these are different. We're going to play spot the difference between these two molecules. I love doing that as a kid and as an adult even. I love to spot the difference between two different things. So, I should have asked you to close your eyes, because now I feel like the secret's kind of been spoiled. What is the difference between these two? Well, it's very subtle, but bam, right there. That is the only difference. And this is why DNA is called deoxyribonucleic acid, is that ribonucleic acid has two OH groups. Here on the bottom of the molecule, we have two OH groups, right? And on DNA, we have one OH group and then one H, right? So compared to RNA, DNA has one less oxygen. So we say DNA is deoxyribonucleic acid. Deoxyribonucleic acid, you know? So we say that, so like I said, we have three portions of a nucleotide, right? We have the phosphate group here. We have the nitrogenous base, which we'll talk about here in a second. And we have the sugar. This sugar is called ribose, right? That's the sugar. So the suffix "-ose", the suffix "-ose" means sugar. Sorry, "-ose" means sugar. So we have ribose, and here on this side we have deoxyribose. Those are the sugars that make up the nucleotides, right? So you have to know the difference between RNA and DNA. You have to know that RNA makes a single strand and DNA makes a double strand, double helix, antiparallel, whatever. One is used for the production of proteins. One is used for the maintenance of genetic information. Right? So RNA and DNA have different structures, different properties, and strangely enough, it's all because one has an OH and one has just an H. Sorry about that. That is the only difference, guys. The only difference between DNA and RNA. There's one more difference, and let's get into that, right? So, nitrogenous bases. When we're talking about DNA, when we have a big strand of DNA, the only thing that changes is this portion. Everything else is going to be the same if we have a strand of DNA. Or if you have a strand of RNA, the only thing that changes is this. And this can be 4 different things for both, right? For the case of RNA, we have A, U, C, and G. Okay? We have adenine, uracil, cytosine, and guanine. And you don't have to know the difference between pyrimidines and purines. That's not necessary, guys. That's not necessary. So for the case of RNA, adenine, uracil, cytosine, guanine. For the case of DNA, DNA has A, T, C, and G. Right? And these are, well, the three of them are the same, right? We have adenine again, thymine, cytosine, and guanine. That's the only difference. U and T. That's it. Uracil and thymine. So, when DNA is running antiparallel, or when RNA is binding to DNA, or whatever, there are certain things called base pair rules, right? So let me look up a picture of DNA. I'm gonna give you 2 Oh, that one's really small. Oof. Wait, hold on. Sorry, I don't know what's going on here. There we go. So I'm gonna give you 2 pictures. One to show you kind of what's going on up close, and one to show you what's going on more generally speaking. Okay, so this is going to be from Khan Academy, because I tried to look up this one image, and it's like a transparent image, and then it would really mess up everything. that's a bit of an exaggeration, but not really. So here we have one strand of DNA, and then we have another. This one's running up, this one's running down, right? So these are those base pair rules that we're talking about here. And on a different scale, we have this is from Khan Academy. We see that we have 1 ring, and then we have 2 rings. So 1 ring and then 2 rings. So basically, right, A and T and C and G, They kind of always pair up together because of this, right? Because DNA always has to maintain, excuse me, that 3-ring wide structure. So that's what we talked about, the difference between pyrimidines and purines. Purines have 2 rings, pyrimidines have 1. A and G are purines, and T and C are pyrimidines. You don't need to know that. What you do need to know, however, are the base pair rules. So, A always pairs with T, and C always pairs with G. And this is the case for DNA. For the case of RNA, A always pairs with U, and C always pairs with G. So this one is DNA, and this one is RNA. And guess what, guys? That's all you gotta know about nucleic acids. That's all you gotta know. The difference between DNA and RNA, and the fact that one has uracil, the other one has thymine. And you gotta know what A, T, C, and G stand for: adenine, thymine, cytosine, guanine, and the other one, uracil, U. Cool? Cool. So, proteins. Proteins are my favorite macromolecule because I think proteins are the most important molecule in our bodies. People study proteins, they study how proteins fold, how proteins are affected by certain medicines. Proteins affect literally everything on how we function. There's proteins in your hair that determine whether or not it's curly or straight. There are proteins in your skin, in your fingernails, proteins that send signals to different cells, proteins that make your muscles grow, proteins literally do everything. They do almost everything. They are so super duper important, and that's why they're my favorite. Right, so a protein will have a basic structure, right? A protein, well, just give me one moment, sorry. Oh my goodness, sorry. So, a protein has a basic structure. C, bonded to C. C on the right has a carboxyl group on it. Okay, well, I'll revisit this. And this C on the left has a amino group. This is a basic amino acid. So, when fully uncharged, we may see an amino acid looks like this. But in our bodies, both of these ends are actually going to be charged. So we have the amino group we talked about earlier, and we have the carboxyl group that we also talked about earlier. Positive, negative. One's gaining an electron, one's losing an electron. Right? Or, sorry, no, that's totally wrong. One's accepting a proton, and one's losing a proton. The acid is losing a proton, the base is accepting a proton, right? And the basic backbone structure of an amino acid is this, right? Every amino acid will have this structure to it, will have this basic backbone to it. The only thing that changes, just like with the nitrogenous base of nucleic acids, the only thing that changes is the R group, right? That's the only thing that changes, guys. So the thing about the R group, though, right? The thing about the R group is that it could change drastically, right? The R group can be separated into 4 different categories: nonpolar, polar, positively charged, positive charge, and negative charge. That R group can fall into one of four categories. And especially if you're a biochem major, this definition may change. You may see it like some of them are just weird and don't fall into a category. Some of them are aromatic. Some of them are nonpolar. Some of them are still polar. And some of them can be charged in solution. Whatever. In this class, these are your four classifications. Some of them can be positively charged. Some of them can be negatively charged. Some of them are polar. Some of them are nonpolar. Right? So, if we take an example, like if we have, let's say, okay, well, let's go ahead and, whoops, sorry. Let's go ahead and delete some of this. So let's say that, and once again with the bond line structure, that's what's going on here. This is what's known as leucine. Now leucine has no OHs. Okay, even better yet, watch this. This is valine. Valine has no OHs, no Ns, no Os. It's just hydrocarbon. Right? So what does that mean? If something is just hydrocarbon, is it polar or is it nonpolar? Well, since you can't answer me, I'm just going to say it is nonpolar. Right? So this would fall into the nonpolar category. Okay, what if we add an OH here? Now, this is known as something called threonine. Now, threonine, because it has the OH attached to it, it is now polar. Right? So these amino acids can change as you go along. If you see them made primarily of hydrocarbons, just say they're nonpolar. If you see one with a positive charge, say it's positively charged and it is a basic amino acid, because basic means positively charged. On the other hand, if you see something with a negative charge, you can say that's acidic, because acids lose that proton and become acidic in solution, right? Or already are acidic and lose the proton, right? And one more thing that I want to emphasize, right, is that this side is the carboxyl group and this side is the amino group. The amino group is positively charged. The carboxyl group is negatively charged. Remember that. You will need to know it. You will need to know it. All right. So, another thing that you need to know. Another neat little thing are the protein foldings. So proteins have four levels of folding. Primary, secondary, tertiary, and quaternary. So primary is the sequence of amino acids in a chain, right? So, like I said, the only thing that changes is the R group, right? But that being said, that R group can change a lot, and the specific sequence of amino acids is very important, just as a foundational level, what amino acid is going to be formed, right? So if you have something like hemoglobin is always going to have those specific amino acids in a specific order, right? Or insulin is a protein hormone. Insulin will always have that same amino acid sequence, right? So secondary. So primary is just amino acid sequence. Secondary structure are what I call backbone interactions. And let me look up an alpha helix so you could see what an alpha helix looks like. And a beta sheet. Let's do that. So secondary structure is when the backbone of an amino acid interacts with itself, right? So look here. We have a double bonded O to the C. Now this double bonded O is going to have a partial negative charge, right? It's just going to have that partial charge. It's going to want to make hydrogen bonds with other stuff. And when you do that, when proteins in these large complex structures make these hydrogen bonds between the backbones, you get this. Alpha helices and beta pleated sheets, or just beta sheets. That is secondary structure. That's as simple as it gets. Alpha helices, beta sheets. These are both secondary structure. These both have to do with just how the hydrogen bonds interact with each other. That's really it, guys. That's secondary structure. Backbone interactions. Alpha helices, beta sheets. It's all secondary, right? So now with the case of tertiary is when we start worrying about this down here, the R groups. So now the R groups are where all the interesting stuff happens, right? So a few things can happen with the R groups, right? If you have a negatively charged amino acid and then a positively charged amino acid, well, opposites attract, so those are going to want to make an ionic bond with each other. Okay, well, how about two amino acids that are nonpolar? Well, so if amino acids are existing in water, if these amino acids, if you have these amino acids that are nonpolar, they're going to want to stick with each other. They're going to want to be together, right? They're going to want to be away from from everything else. They're going to want to stick together, the nonpolar ones, right? And I know I was ranking on how much I hate sulfhydryl. When you do have a sulfhydryl group here, when you do have a sulfhydryl group in an amino acid, it can create something called a disulfide bridge, which is a covalent bond, and the only example of a covalent bond in an amino acid. You don't need to know that. It's just something to mention. That's all tertiary, guys. Tertiary is the interaction of R groups, right? So primary is amino acid sequence. Secondary is backbone interactions, aka alpha helices, beta sheets. Tertiary is R group interactions, so that could be anywhere from covalent to nonpolar interactions to polar to ionic. That's tertiary. And quaternary is when you have two amino acid chains, or two or more amino acid chains, like two completed amino acid chains, and they come together to make one big protein. So quaternary would be like protein interactions. So let me pull up an image of hemoglobin. That's like the number one example I can think of when I hear quaternary structure. Bam. That's hemoglobin. So you see you have the blue sections and the red sections. They're all different. They all contain their own little subunits. Right? No. They are their own subunits. Each different portion are their own subunits. And I think that's something important, right? They're just the protein interactions. You don't need to know quaternary super in depth, but I think it's worth noting. So now, proteins have a certain thing about them, right? So now proteins exist So proteins exist at a specific pH, at a specific heat and temperature, and at a specific folding, right? So heat, you've just been kind of looking at a bunch of nothing for the past 5 or so minutes. Heat and temperature are both super duper important for proteins, right? Because once we start, I just realized that those are the same thing. Sorry. Heat and acidity. So heat and acidity are super important for proteins, right? If we crank up the heat too high, right, this is why a fever is very bad in our bodies. If you crank up the heat too high, then those hydrogen bonds made in secondary and tertiary structures start to break apart, right? Very bad. And when we crank up the acidity or crank down the acidity, if we make something more acidic, less acidic, more basic, less basic, if we're changing the pH of the solution that an amino acid lives in, you could change how that amino acid folds, right? So if something is so acidic that if something is so acidic that sorry that an acid regains a proton, then it's no longer going to be able to form an ionic bond, right? Or if it's something so basic that if it's something so basic that sorry that a basic amino acid loses its extra proton, then, like I said, ionic bond cannot happen again. So when this happens, when the structure of the amino acid is compromised, this is known as denaturation, or just denaturing. When a protein is denatured, its function is kaput, right? Its function goes away. It's no longer useful. It no longer is able to perform its function properly, which is why cranking up the heat or acidity or temperature or pH of something of your body, it's super bad for your proteins. It's super duper bad. So that's called denaturation, or just denaturing. So, yeah, we went over the three. So let's just go over some quick trivia. Maybe not trivia, but let's go over some quick notes. Carbohydrates. The monomer of carbohydrates would be just monosaccharides. The bond formed between carbohydrates would be a glycosidic linkage. And the function is energy storage, or structure. That's carbohydrates. Nucleic acids, the monomer is nucleotides. The bond, and pay attention to this, is a phosphodiester linkage. Quite a mouthful. And the function is for genetic information / utilization. Proteins, the monomer are amino acids. The bond, and once again, pay attention to this, is a peptide bond / linkage. And the function is pretty much everything. They have so many different functions. Proteins have so many different functions, including energy. usage. So these are kind of our big three, right? Where they have monomers, they have a specific type of bond, and they just kind of have that sort of thing. They have that thing going on about them, right? So the thing about lipids is I just realized this entire time, this is humiliating. I spelled lipids incorrectly. That's oh, my apologies. Lipids, not liptids. Lipids. Sorry, ignore that. Lipids is our last topic for the night. I've been going on for about an hour and a half, so we're going to stop here with lipids. There are three types of lipids, or like three subcategories of lipids that we like to know about. So, triglycerides, phospholipids, and steroids. So that's the ones that we care about. So when we talk about a lipid, right, it's important to know what a fatty acid tail is. Right? So a fatty acid is essentially just this. So we have a carboxyl group at the end, right? So that's where it makes the bond. And then we just go on and on and on and on and on and on. This is the fatty acid tail. Right? What makes it fatty is that it is mostly hydrocarbon. Like past this point, it is all hydrocarbon. Right? So it is a very nonpolar molecule, therefore. So this is a fatty acid. So remember what I said earlier, when we have an apex, or if we have something at the end, that tells us that there's a carbon with its associated hydrogens. So it takes way too long to draw out a fatty acid with all the carbons and hydrogens. So I like to shorthand with the bond line structure that I've shown you here. Right? So, sorry, just 1 second. So the thing about fatty acids, right, is that fatty acids can be either saturated or unsaturated. So what this means is, let me pull up a good example. You've probably seen this image before. Whoops. Sorry. One moment. So here we have saturated versus unsaturated fatty acids, right? So here you actually see it drawn out, like all the carbons and hydrogens. So we have the carboxyl group here, right on the ends. But the difference between saturated and unsaturated is that saturated has the maximum number of hydrogens bonded to carbons as possible, right? So in other words, it's saturated with hydrogens, the molecule is. Versus unsaturated, notice this double bond, right? So because of the presence of the double bond, there is no, the molecule does not have the maximum number of hydrogens bonded to carbons. It is unsaturated, therefore. And in this class, it kind of works on a binary, right? Either something is saturated or it's unsaturated. As soon as we see this fatty acid tail with the double bond, we know that is unsaturated. So why do we care, right? So let me look up an example of oleic acid. Maybe not oleic acid. Maybe not. No, that's not a good example. So here, like, I just want to avoid having to draw this out because it is a lot. It is quite a lot. Yeah, that should be good enough. Okay. So this is a triglyceride. So this is one of the three that I was talking about. So this is a triglyceride, right? So tri meaning three, glyceride meaning referring to the fact that we have a glycerol backbone, right? So I gave this example to a student the other day. How the glycerol, if you ever played with a Mr. Potato Head, you know that the glycerol is kind of like the potato, and then you can kind of add whatever you want to the glycerol. So in this case, for a triglyceride, we're adding 3 fatty acids, right? So we have 1 fatty acid, 2 fatty acids, and 3. So just judging from this picture, which one of these are saturated and which one of them are unsaturated? And pause the video if you would like, but this one here in the bottom is unsaturated because we have the double bond here, right? So why do we care if something is unsaturated versus saturated? Well, when something is unsaturated, notice how in the case of these two, they just kind of go in a straight line, versus the one with the double bond, it kind of goes in a straight line and then makes like a sharp turn, right? When we have these kinks in fatty acids, they become less dense, right? They become less tightly packed together because they kind of have an arm sticking out, like that makes it harder to bond with, right? So you have something like butter. Something like butter is solid at room temperature. Something that's butter will primarily be made of these saturated fatty acids, right? Because saturated fatty acids can be tightly packed closer together versus unsaturated, which cannot be. So something like olive oil or peanut oil will have primarily these double bonds, these unsaturated fatty acids, because it is liquid at room temperature and has these kinks that stick out and are able to, have a lower ability to bond closely together, making it less dense, making it liquid, right? So that's the difference between saturated versus unsaturated. So do you remember how I said that phosphorus will come in handy later? Well, now we're talking about phospholipids, right? So the cool thing about phospholipids is that, like I said, we take that glycerol backbone, and instead of 3 fatty acids, now we just have 2, right? So I'm going to shorthand a phospholipid to just a P, and then a tail that's saturated because it's straight, and then a tail that's kinked, and that's unsaturated. So these are your 2 fatty acids, and this is your phosphate head. So phosphate is negatively charged, of course. So this molecule would be considered amphipathic because it has affinity for both polar and for nonpolar molecules, right? So, like I said, if we have these 2 tails, and the phosphate, that's a phospholipid. Specifically, if we have the phosphate, listen closely, if we have the phosphate, it's a phospholipid. I hope I made that clear enough. So, moving on. That's a phospholipid, and we'll get into how important phospholipids are in the next exam review when we talk about membranes, right? Just know that phospholipids are amphipathic, have two tails, and a head that's hydrophilic, and the tails are hydrophobic, right? So, yeah. Let's talk about our last thing. Steroids. Steroids are pretty cool because steroids act as a cell signaler. Just like proteins, steroids can act as hormones, right? I mean, think of the example of testosterone, estrogen, estradiol. Steroids act as cell signalers, just like proteins do. And remember that. Proteins act as cell signalers, steroids act as cell signalers. Proteins can act as cell signalers, lipids can act as cell signalers. Remember that. So, a steroid will have a general structure of 4 rings. So when we have these sort of 3 hexagons, plus the 1 octagon, what the hell am I talking about? Plus the 1 pentagon. This is the basic structure of a steroid. Now, in the case of like testosterone, we might have like a double bonded O down here, or in the case of estrogen, it might be a OH. In the case of cholesterol, which is the basis for most steroids in our bodies, we have like a big old like chain with like I don't even know, just big old chain here on the end. Just know that let me make it even clearer, actually. Yeah, how's that, right? Looks like shit, but sorry, excuse my language. Just know that when we have this 4-ring structure, that is a steroid. Like, you don't necessarily have to know what a steroid does. You just have to know that hormones can be lipid in the form of a steroid, right? Or a protein, right? So a good example of that would be estrogen versus something like, I just talked about it earlier. Insulin. Yeah, insulin would be a protein. Estrogen would be an example of a steroid, right? So it's also important that you need to know the general structure of a steroid. Just the fact that it is a lipid, it's still a lipid because all of these rings are hydrocarbons, and they're hydrocarbon heavy, and they're all hydrophobic, meaning that they are going to be lipids. They're going to fall under that lipid category. So, sorry for the flashbang. Okay, so that brings us to the end, right? I hope this was helpful. I hope that I was able to answer any questions that you may have had, because everything that I covered today is going to be on the exam tomorrow. And I mean pretty much everything. I mean, the one-off comments that I made about like certain things, like I can't even remember at this point. It's been a little bit. Just know that most everything is going to be on your exam tomorrow. So I wish you all best of luck. Have a good night. Watch this at 2 times speed. I know it's at the end. I don't know why I'm saying this at the end. Whatever. Take care. Have a good night.